So thank you to Marie for the generous introduction. Actually, I did spend a lot of time at the Salk Institute, which was at the time ranked #1 in neuroscience by objective measures, and I knew the Nobel Prize winning Crick and spent some time with him at the time. And although he was tremendously busy, at 80 years of age, he didn't have time to talk to you because he was trying to work out how the brain was going to make consciousness, and he was too busy to talk. But one of the things he said, he was our work in particular, he commented on at the time, is going to be where the future lies. And that was, as we were at the Salk at that time, dissecting out the very specific circuits, mechanisms, and nerve cells that are involved in things like memory and learning, using very, very sophisticated, high-end technologies that allowed us to manipulate single nerve cells with modern molecular biology. It was really the beginning of an incredible time in neuroscience. And in a sense, today, when I talk about Alzheimer's, I will give, as part of my talk, a bit of a sense of where we are in history. And I hope the message that I want to give will come across, and that is that whilst it could be very frustrating for someone who's diagnosed today with something like dementia or Parkinson's disease, the fact is, and thankfully, as a result of scientific progress and scientific effort, we can say to you that with 100% certainty, we will have treatments for these diseases. It's a matter of when, and it's not a matter of if, and it certainly is a matter of public support and government support for what we do, because without that, you won't have the scientists doing what we do. So, the problem is extensive. As Marie said, I'm going to focus today on dementia, and as part of that, Alzheimer's being the most common form of dementia. But the extent of this problem is staggering, and it's a call of arms. We have a problem a society faces that requires grave attention and very serious effort to overcome. And this is starting to be recognised by federal governments around the world, but I think the extent of it is still not apparent as we see people living longer, and as people live longer, diseases that used to be diseases of aging but become diseases of middle age, I'll clarify that in a minute, are going to be very increasingly serious. So 1,800 new cases every week of dementia, that's only in Australia. Every 6 minutes, someone new is diagnosed with dementia. Today, 350,000 people approximately have dementia in Australia, and that's just the people with the dementia. As we all know, they've got families, nurses, carers. There's a huge impact of those 350,000 people, and we all know people who've been affected and who are directly affected by their families, having it in their family, because they've got to be there to care and to look after and to worry about their loved ones. And the statistics are that if there's no major medical breakthrough, and as you just heard, I believe there will be medical breakthroughs, but without a major medical breakthrough, there's going to be almost 1 million people by 2050. It sounds like a long way away, but I'm sure people will remember the year 2000. It wasn't so long ago. 2050 approaches very, very quickly, and if we don't solve this problem, we've got almost 1 million people directly affected in Australia, without mentioning the loved ones, the families, and the carers. And it's the third leading cause of death, unfortunately, in Australia and worldwide, and the costs are very significant. We're talking within 20 years for the federal government, 1% of GDP, which is a huge amount of money, is going to be dedicated to treating and caring for people with dementia. And so the message today is, unfortunately, there are no cures. There's nothing that you can give to someone and say, look, here's the medication, go to the doctor, get the medication, and you'll be able to slow the disease or prevent the underlying disease from progression. But the fact is, as I just said, there is tremendous hope, and that hope is within science. And I'm sure all of you remember, maybe it was 20 years ago, you went to the doctor and you had high blood pressure. It's called hypertension, but high blood pressure. The doctor had some last-line drugs, some drugs that were not very ideal because they had terrible side effects. And most of the time, the doctor would say to you, don't get so stressed, you need to reduce stress in your life. And as we all know, if someone tells you not to get stressed, what happens? You get stressed. I'm not stressed! So there's that problem. And then they would tell you to exercise and to change your diet and to take less salt. If you go to the doctor today, I don't think they tell you not to get less stressed, because it's something that's very hard to control. We all get stressed. They probably will still tell you not to take so much salt, but they'll give you a medication, and that's where we want to be with dementia. We want to get to a point where we have a disease-modifying therapy, a therapy that prevents the underlying problem and prevents the ongoing progression of the disease. And in fact, if you think about how far medical research has come, and that's what I think is the main message I want to give today. If you walked into a pharmacy in 1939, there was nothing, really. The first antibiotic, penicillin, was discovered, or actually came online in 1939, and the first person who took it, definitely the infection went away, but they ran out of it because they had so little, and they tried to rescue it from his urine, they couldn't get enough of it back, and the person eventually died. And such is the case of most medical research, that initially the first discovery is given in small amounts, and it's given with great risks and very little success, and then slowly we get to the big outcome. So the big discovery of antibiotics, and here we are today. You go to the doctor, and you expect that you should be out of the doctor, be given a prescription, go to the pharmacy, and be given a therapy. And you will find numbers and numbers. If you go to the average pharmacy now, you know the shelves are full of drugs that are available for numerous conditions. And that is the success of science. That is because there are scientists working away in laboratories around the world trying to understand disease and trying to come up with an understanding that allows the development of drugs by the big pharmaceutical companies. And the success is self-evident from the fact that people are living a lot longer, and they're living a lot longer because we've done great things with cancer, we've done great things with blood pressure, and I can assure you the scientists now there's also scientists beavering away trying to understand Alzheimer's and trying to come up with the right answers, and it's through that work that we're going to have drugs on the shelves that are going to help people with dementia. The problem we face, though, is the complexity of the brain. And this really, in fact, the extent to which we have made progress for dementia and that we have made progress for Parkinson's sometimes astounds me as a scientist, because I will also say that it's an incredibly wonderful time to be a scientist. It's a period in which it's a great honor to be a scientist, because we are really making such progress, and we are in what I call the equivalent of the Industrial Revolution in medical research. We are making tremendous progress, and it's fun to be doing science as well as a responsibility. But the brain is a great mystery, and its complexity is very, very hard to unravel. We know a bit about the brain in terms of its overall structure, because as you can see here is a picture of the brain. Just point to this here, if I can. Whoops, I've gone backwards. I'm not sure how that happened. Anyway, on your left is the front of the brain, and on your right is the back of the brain. And you can see the bits that are colored basically points to sections of the brain that do different things. And we know that because if you have damage to those regions of the brain, those functions are lost or damaged. Okay, so deep in the middle of your brain is a section that controls memory, and that's called the hippocampus. But there's lots of other things involved in memory. And we know that from structural studies and from damage to the brain and from increasingly sophisticated studies. At the other end, we've got this understanding that the brain is made up of these things called nerve cells, which are submicroscopic. You have to look down a microscope and you can see nerve cells, and you can actually get them out of a brain and put them into a culture dish, and you can study them. But to understand how there's 100 billion nerve cells that make up this brain structure. And to try and understand how those nerve cells, together with the other nerve cells in the brain, bring about our brain functions is so beyond us at the moment. We're so beyond our capabilities that it's an enormous challenge. And as I said, it's actually fun as a scientist and exciting and interesting, but it's got real implications for people who have these diseases, because we need to understand it ultimately in order to be able to treat some of these diseases. But having said that, as I said, we're in the middle of a revolution, and I believe we are making tremendous progress through some of the major advances that I will talk briefly about today. So to give you a perspective of Alzheimer's and where we are at, just a very brief timeline of the history of that and the science of it. So again, to say how frustrating it is today for someone who's diagnosed with dementia or the families, but to say this is how fast things are moving. So in 1906, Alois Alzheimer described Alzheimer's disease, and the reason he described it was he had a patient, a 51-year-old woman, who had memory problems and some disturbances of behavior, and when she died, he took her brain and analyzed it. Now, this is how science works. Many people don't know this, but just down the corridor from Alois Alzheimer was a guy called Nissl. And Nissl had invented a brand new way of looking at staining the brain, and it was called the Nissl stain. And we use the Nissl stain today in our laboratory, as do many people around the world. It allows us to actually look at structures in the brain. So, as happens at the Garvin sometimes, where we run into people on the stairway and we find out something interesting they're doing and we talk about it, and leads to very unexpected research advances. And that's why it's important to be at a place like the Garvin, where there are such fantastic scientists that we interact with and we share our information with and we discuss our ideas with. But Alzheimer did that 100 years ago, more than 100 years ago, walked down the corridor and chatted to Nissl and said, look, I'd like to look at these brains. Can we try your stain? And we'll look. And he looked down the brain, down the microscope at the brain of this woman, and she had these substances which we now call plaques in the brain. These substances which, it's called plaque because it literally looks like plaque down a microscope, like tooth plaque. And very shortly after that, the disease became and was called Alzheimer's disease after the person who diagnosed and discovered it based on those plaques. But then not a lot happened until 1976, when Alzheimer's really became called the most common form of dementia. And then now in 2012, we've come to the point where we understand that, in fact, dementia begins almost 25 years before you actually see symptoms in a person. So unfortunately, people who get dementia are starting to get it in their 60s, possibly even earlier. And that's a very frightening piece of information. But looking at the timeline, and I'll take you through it pretty quickly, but I could go on about each of these developments in detail, but I don't have the time. But 1968, the first cognitive test, the first test for memory that was used to start to objectively diagnose dementia. In 1984, an Australian discovery, and Australians have been profoundly important in Alzheimer's research. In 1984, the discovery that the substance that makes up the plaque is a substance called amyloid beta it's called. And amyloid has been, at 1984, that was done by a guy called Colin Masters in Melbourne, an Australian, who discovered this substance and said that he thinks it causes Alzheimer's. Now, as happens in science at the time, no one really believed him, and it wasn't until a couple of years later that someone else isolated the gene that often can cause Alzheimer's and showed that, in fact, amyloid probably was the cause of some Alzheimer's disease. And as I'll say, I don't think it's the cause of all Alzheimer's, but some Alzheimer's. Following that came the discovery of a protein called tau, which, as I explained, is the substance that's thought to make up tangles in the brain of Alzheimer's. And then the first drug for Alzheimer's, which actually came out of a sort of an idea that nicotine, which was in cigarettes, was supposed to be protective, which actually we don't believe anymore about cigarettes. But nicotine, as a result of that discovery, there was thought that we should increase the substance in the brain called acetylcholine. It doesn't matter. An Australian, came up with the discovery of a drug called Tacrin, which has now been replaced by more modern drugs. And he was working in the United States at the time, and he came up with the first drug for dementia, which helps with the attention and helps a little bit with memory, but it doesn't, as I said, alter the underlying cause. But again, an Australian major discovery, one of the big breakthroughs. First Alzheimer's modifier gene in 1993, and in 1995, where things really started to heat up, was a mouse model of Alzheimer's. Now, mouse models in science are very, very important. We can create or replicate aspects of the human condition in mice, and then we can try and get the mice better. And to the extent that we can get the mice better, predicts the extent to which we might be successful in people. So the first model was announced in 1995, and in 1999 they treated the first mouse with a vaccine which was successful. That was 1995 to 1999. It was incredibly fast. Unfortunately, in 2002, it was announced that failed in human beings. Okay, so that was 2002, the first drug of significance tried in humans, and it failed. It's only 2002, and here we are in 2015. So that was what I call incredibly fast, from identifying the gene to getting the first drug to where we are today. So where are we today? So, as I said, there are many hallmarks, but there are two major, what we call hallmarks of dementia or of Alzheimer's disease. And if you look in the brain of a normal person, especially the memory center of a normal person, it's quite a nice, beautifully organized structure with nerve cells and the surrounding other kinds of cells which play an important role. These cells are called glia, it doesn't really matter. But you can cut open a brain, and it's remarkable to the extent to which brains look incredibly similar. I find this remarkable that we all develop so similarly. But if you go into a person who's had Alzheimer's disease, or even the mouse models of Alzheimer's, you see quite a different picture. And that is, you see these substances called plaques, which, as I said, almost look like tooth plaque down a microscope. And another substance called tangles, which are as they sound. They're tangles in the brain, and they are associated with neurons and the nerve cells in the brain. And these tangles are made up of substances called tau and the plaque is made up of a substance called amyloid beta. Okay. So in terms of existing therapies, and I'll come back to how we going forward in a second, but just to sort of bring you up to speed of where we are today. There are some medications. If a person has to go today and get diagnosed with dementia or Alzheimer's, there are some therapies that are given. These are drugs like Aricept and another drug called memantine, or which is known as Bixa. And these drugs provide some benefit to some people, but the effect, unfortunately, is not profound. And as I said, they don't seem to affect the underlying condition. So they might provide some better cognition for some period, but they don't stop the eventual decline, very unfortunately. There are treatments that are given for the behavioral changes, for depression, for anxiety, and there are treatments for the sleep changes that occur as people go into dementia. But these drugs, again, don't really cure the underlying problem, and there are big questions at the moment to the extent to which we should be giving drugs for behavioral changes and for sleep changes, and the extent to which we balance up the help that they give against the damage that they can do. So where to? So where do we go and try and find the answers? So a lot of the current world research is focused on these two substances I've mentioned a couple of times. One is called amyloid, which makes up these plaques. So when Alzheimer discovered these plaques in the brain, it's been thought for many, many years that these substances that makes up these plaques, not the plaques themselves, but the substances that makes up those plaques, called amyloid, might be causing the disease. And there's good evidence for that, but there's also evidence against it. So there's been enormous effort worldwide in trying to block the amyloid. Enormous effort. Billions and billions of dollars spent by drug companies. And unfortunately and very sadly, the news is that those drugs aren't succeeding at the moment. In fact, just in 2014, 4 big international trials of 4 brand new drugs, each costing about $1 billion each to develop, failed in clinical trials. So this is very bad news for society and also for the drug companies. I mean, people often think of these drug companies as these big behemoths that are evil players in the world, but actually the contrary is kind of true. Yes, they have to work for profit, but at the same time, they do invest the $1 billion in the research. And if you do that a few times, it starts to eat into your bottom line and actually threatens the company's survival. So there are serious problems at the moment in terms of how do we take these kind of big trials forward and develop these drugs. And to continue to rely on the drug companies alone is really going to be a problem. And in fact, what's happening now is the drug companies are pulling the resources together and pulling resources with government and with basic scientists to try and come over these problems. And I would say the drug companies are doing a wonderful job in trying to get there, but it's a big, big challenge. So the amyloid story at the moment, the idea that we can block amyloid, remove this substance from the brain, is not looking as promising as we hoped in all cases. It might still work in some cases. So as a result of that, the anti-amyloid therapies, as they're called, are still in progress and people are still trying to work on them. And I'll come back to that briefly. The other target that is being targeted, the other approach that is being developed more recently is to try and remove the substance called tau, which makes up those tangles that I talked about. And there are efforts now that are going on around the world to try and block this tau. Again, the basic science is done by the basic scientists, like those in Australia who discovered these causes of Alzheimer's, like what we do at the Garvan. We do the basic research, we come up with the basic understandings, then the drug companies use those basic understandings to try and target drugs or develop drugs that block the problem, right? So one of the problems that's been identified by basic scientists is this substance called tau. The drug companies try and come up with substances called, or drugs called anti-tau, and they are trialing those at the moment to see if those will help. And the jury's out. The trials have to be done, again, at enormous cost to these drug companies and at enormous cost also to society. So whilst those two directions are continuing and there is increasing debate about their validity because of these recent failures, science is not put off, and in fact rigorous debate and rigorous. Controversy is part of the scientific makeup. It's what we do. When two scientists are arguing together, it doesn't mean we're fundamentally disliking each other or we're at loggerheads with each other personally. It just means that we're trying to get to the truth. And scientists do debate rigorously and vigorously as to whether their idea is more right than the next person, and continue to accrue data and evidence that goes with their idea or against their idea. And eventually we see how science comes to the right answer. And I love the discovery that stomach ulcers were caused by a bacteria by a Nobel Prize-winning Australian who was made fun of for many, many years, until it was proven that in fact it was bacteria that caused stomach ulcers. And so there was a debate for 10, 15 years about whether they had any validity, but the nice thing in science was the truth prevailed, and the Nobel Prize, which is the highest honor, is given to the person who got it right, even though they were made fun of, in a sense, at the beginning. Science is made up and needs to have different ideas. So where are the emerging directions? There's been a lot of talk about stem cells and regeneration. There's a lot of talk about something called inflammation in the brain, and there's a lot of talk about other targets. And unfortunately, I don't have time to go into all of those today, but other than to say that there are coming up a lot of other ideas about what causes dementia and ways that we can modify that. So, as I said, the idea of dementia being caused by A beta, is it wrong or incomplete? There's vigorous debate at the moment. There's a lot of people who are saying, look, it's still right, we just haven't got the therapy right yet, we need to give it to people earlier. So despite the fact we had big failures last year, it might be that we're giving it to the wrong population of people. We might need to give these drugs to people earlier in the disease. And then the problem, and given that the disease starts 25 years before we actually see the symptoms, maybe we need to be able to try and give this to people many years before we actually see the symptoms, okay? Then, and how do we do that? Well, we have to identify approaches to diagnose dementia before we see the memory problems. And so you'll be seeing in the media at the moment a lot of announcements of people who say they've got better and smarter and nicer ways to diagnose dementia. And that's because scientists are saying we need to be able to identify this disease early, we need to identify it before the memory problems, and we need to have clever ways to do that so that we can try and give some of these drugs earlier. And if we can give these drugs earlier, then we'll be able to see they actually work, right? So there's a lot of effort going on in the scientific world at the moment to find better ways and earlier ways to diagnose dementia, and then to trial these drugs again in people earlier on the hope that maybe that will actually prevent the disease. If we can remove this amyloid earlier, we get it before the horse has bolted, so to speak. But there's also a growing thought that maybe this whole amyloid idea is wrong, and consequently, as I said, there's this beautiful sense of emerging alternative ideas, and many scientists coming out of the woodwork saying, look, I've got other ideas about what might be causing Alzheimer's, and I'd like to think that we're one of those groups coming up with alternative ideas about what might be causing dementia. Unfortunately, in today's talk, I don't have time to go into our specific work, but it's needless to say something I'm very, very excited about at the moment. So in terms of tau, where we're at with that, it looks like tau, at least in terms of diagnosing the disease, markers of tau, so the aim of scientists at the moment is to give a substance in the blood that goes into the brain and marks these tangles, and then you can see it with something called positron emission tomography, or PET scanning. And the idea at the moment is that we can give these dyes to a person and then analyze their brain and be able to diagnose them with dementia a lot earlier than we currently are doing. And that is looking very promising in terms of as a diagnostic. And as I said, the other thing that's coming out at the moment is this idea that we might be able to give drugs that block the tau, and that's in clinical trials at the moment. So we stay waiting for that, and we'll see how that progresses. So in the meantime, in the absence of great drugs, what do we have at the moment? So there are things that can be done today to try and reduce the risk of dementia. And in fact, as is always the case in science, some of these discoveries about what are affecting the risk of dementia also are giving us clues and ideas about ways that we might be able to treat dementia, right? So what are those things? So there's diabetes. It's now recognized that a person with diabetes has a much higher risk of dementia. So there's a good argument these days for trying to reduce maintain or control diabetes very, very well. And that's becoming an increasing story within dementia. High cholesterol and high blood pressure. In fact, the things that are classically associated with blood vessel disease. are also risk factors for dementia. Obesity, as I'll show you, is a major risk factor. As with everything, carrying too much weight provides great risk for the brain. Alcohol is this idea that we should have half a glass of wine every day, and I get asked this regularly, should we have half a glass of wine every day? Well, there's two answers to that. I don't know how many people actually stop at their half a glass. And the second answer is that it looks now that research is saying, look, maybe half a glass, but if you take it, but that's only if you take it before you have any signs of dementia. But once a person has some signs of memory loss, any amount of alcohol actually increases the dementia decline, the rate of decline. So alcohol in general, not highly recommended. Coffee was one of the surprises, because we've gone through, as happens in science, quite a few debates. There was a time when coffee was bad, and there's a time when coffee is good. And many of you in the audience probably wonder what's going on in science, that they can have one answer one day and another answer another day. And that's, again, the nature of science, that we get a lot better at what we do. We have a lot of people doing a lot of studies, and we have better statistical techniques, better ways of analyzing data. We just get better at doing science as time progresses, and we get better at analyzing lots and lots of data. And as a consequence, we get closer to the truth in time. And at the moment, the idea is that coffee is actually probably, may even be slightly helpful, but it's certainly not harmful to be drinking some coffee. Certainly lack of sleep, unfortunately, has been recognized as a risk factor for dementia. If a person's not getting enough sleep, it certainly is a sign that they're at greater risk of dementia, but it's not clear if that's a cause or a consequence. We certainly know that as the brain gets damaged, it's harder for the brain to actually put itself to sleep, right? So we don't know if it's because the person is beginning to get dementia that their sleep patterns might be disrupted, or if in fact it is the fact that as you sleep less, you are at greater risk of dementia. We don't fully understand that yet. There's certainly evidence that air pollution, maybe even pesticides, increase risk. And then there's something in the blood which is called homocysteine, which is known now also to be a risk factor for dementia. How am I going for time? A couple minutes? Okay. And then meanwhile, research also says that a Mediterranean diet, the classic Mediterranean diet, that is diet of healthy fish and vegetables, olive oil, seems to be quite protective. Whole grains, not processed grains, low dairy, nuts and legumes, okay? And that seems to be holding up quite clearly, that what you eat can affect your brain. Vitamin B tablets, just a good old vitamin B from the local pharmacy, a multivitamin B, contains some of the essential Bs that seem to be helpful for the brain. And there's increasing evidence, and I think it's indisputable at this point because the evidence is so strong, that exercise, and we're told this for everything, right? Exercise is really very important for the brain. And it's just a case of actually getting a good walk, a good solid walk, a little bit of a decent pace of walking, and this can actually help with the dementia risk. Now, of course, that's a hard thing to often tell people who are getting older, because, of course, walking and exercising is a increasingly hard thing to do as a person gets older. And so one thing that we're saying more and more is that you've really got to get into the exercise habit when you're a bit younger. Certainly, if you can do it when you're really young, is ideal. And I think there should be a lot more pressure on kids to be getting into exercise, regular exercise. But even in their 50s and 60s, when you're young in your 50s and 60s, should be a time when you're really getting stuck into exercise so that you can keep your body fit for later in your life. And I think that was the point I wanted to make, and that is why dementia is so much increasing, right? Is that we used to have the old age pension, it was called, for people who reach 65, and they might live another 3 or 4 years on the age pension, and unfortunately would pass away as an old person in their 70s. But nowadays, thankfully, we think of 65 as middle age, and 70 as still reasonably young. And that's, again, medical research. So I believe dementia is actually a middle-aged disease these days. I don't think we have time to go through all. I've got half a minute. Okay, so I was going to take you through the evidence, and what I was going to show you was that, you know, there's a lot of evidence for the statements I've just made in the scientific literature, and the evidence is substantial. But even to show you also that something like diabetes, there were numerous studies, and each of those studies gave slightly different results. And that was a point I wanted to make, that science progresses slower than we would like, but it progresses. And there were different results about what diabetes risk factor is for dementia. But at the end of the day, when people come together and analyse all that data together, it became absolutely clear that diabetes is a risk factor. In terms of our research, I'll leave it on that note. To keep it really, really broad, but what we do is very sophisticated, high-end technologies that the Garvan Institute provides. And I can't stress again how important it is to be at a place like the Garvan, where we get access to excellent colleagues, front-end technologies, incredible genomic resources, incredible microscopy resources that allow us to do things that really are cutting edge on the world stage. And to say again that, you know, Australians contribute massively to world research. You often get the question, well, what can Australians actually really do? Why don't we just leave it to the United States, who've got the real money? And the answer is twofold. The answer is it's not true. Australia does incredibly important research and contributes massively. And secondly, one of the things that, because we contribute so well, we get invited to major meetings and major discussions in the United States or anywhere in the world, and we get access to the world-leading research that's happening everywhere, and we get to bring that research back to Australia and to share it with government and to share it with medical doctors and to make sure leading research comes into Australia. Anyway, I think at this point, I'll just say that our goal is to do two things. If you think of dementia as a disease of neuronal loss, our goal is to try and replace the neurons that are lost using a process called regeneration and to block the nerve cell death. And we've got some specific strategies that we're working on to achieve that. So I'll leave that slide up. That's where Garvan and our research is heading. You can have a read of that, but I'll leave it at that point.
Thank you very much, Bryce. Look, I know many of you do have questions, but we did field questions ahead of time just to make sure that we could just kind of wrap up each session kind of efficiently. And so I do have a couple of questions that have come from the participants in the audience for Bryce that I'd like to ask him. And one of them, it does touch on, or both of them do touch on the plaques that he's talked about. And the question is, really, somebody's asked about predictive tests based on proxy plaques forming in other parts of the body because it's difficult to analyse the brain. So, for example, in the blood vessels in the eye, there is a study showing that you could fluorescently label plaque deposits in the eye that are similar to Alzheimer's. So, is that a useful predictive model for marker for Alzheimer's?
Discovery and something, again, fantastic that Australians are doing it. I'll just reiterate what I said in the talk. At the moment, science is in a kind of a uncertain situation on the plaque situation. So what the idea here is, what Marie's talking about, is this idea that if we can see the plaques early, before we see the dementia, then we can give drugs early, and that's a really important idea. So there's a lot of scientists who still think plaque, or the substance that make up plaque, amyloid beta, is the cause of disease, and if we can remove it, we're going to stop the disease. So they're trying to find ways to diagnose it easily, and one of those is to look for plaque in the eye. But that goes to beyond that is the question of whether the plaque is actually going to be predictive, and the jury is out. So whether this test is going to hold up over time depends on whether plaque actually is a predictor of disease, and at the moment it's highly controversial and is unclear.
What about another question on plaques then, Bryce? Because I'm a plaque favour. I favour plaques. Don't know anything about it, but I do somehow favour plaques. There's a study just been done in Queensland showing that if they use ultrasound to break up the plaques in the brain, that's good.
Right, so, right. To understand the difference between correlation and causation. So what was interesting about that study was not that the plaques were disappearing, but that the memory, it was done in mice, and what they did in those mice was to give this ultrasound to the brain together with what's called microbubbles. Now, microbubbles also contain oxygen within them, and it's not clear exactly how this was working. But the idea was in the mice, it actually helped the memory in the animals. So that was what was interesting. But it's kind of a circular argument because these animals are engineered to produce more amyloid. And so in those cases, there's no question that amyloid causes the disease. And certainly there are some humans who make too much amyloid and that causes the disease. But I'm just saying in a certain group of people, amyloid may be the cause of disease. But what was interesting about this study, regardless of that argument, was that the memory was restored in these animals as a result of this ultrasound treatment. it's going to be a big question as to how well this goes forward into humans. It's a long way road ahead, but I really commend, it was Jürgen Gotz in Queensland. He's a colleague and a friend of mine, and I just commend his work, and I was really, really excited to see that discovery. And whether the plaque being removed was the reason it got better, I can't tell you, but the fact was the memory got better, and that's what we care about.
Very much, Bryce. And so you can see that there's a whole lot more research on all different fronts that is required to be done by Bryce and his colleagues and other people around the world. So hope to see you in 10 years' time, and we might have the answer. Thanks, Bryce.